Explore your knowledge with the Botnica.
Hello friends, our new topic is metagenomic and microbiome engineering. So before we dealing with metagenomics and microbiome engineering, we have to know this about this basic terminologies, that is what is genome, what is genomics. So genome in molecular biology and genetics, a genome is defined as the genetic material of an organism that contain either DNA or RNA and the genetic material of mitochondria and chloroplast. So in simple words, genome is a genetic material of particular organism. It either DNA or RNA. These are called as genome. Genetic material of an organism called as genome. So next terminology is what is genomics? The study of complete set of DNA in a person or other organism. The main aim of genomics is to study the entire genome. Genomics is the method depend on the preparation of pure culture, and as a result, this method have limitation in studying non-cultivated organism. Genomics is an interdisciplinary science which focuses on the structure of a gene, mapping, evolution, and function of the gene. So listen, genomics, it is the study of a genetic material of a particular organism or the study of their genome. It is called as genomics. The main aim of genomics is to study the entire genome. In genomics, we study the structure of the gene or their entire DNA sequences. Genomics method depend on the preparation of a pure culture. So this genomics, this is a method to study the genetic sequences or to study the genetic material of the particular organism, but it is depend on the preparation of pure culture, and as a result this method have limitations in studying non-cultivated organism genome. We are unable to study genome of non-cultivated organism. Okay, so next is genomics is a interdisciplinary science which focuses on the structure of the gene, mapping and evolution and function of the gene. In genomic method, we study the structure of particular gene, and also we study the gene mapping and evolution of particular gene over a time period, and also the function of the gene, which gene express which character. These all studies of the gene related to gene or related to genetic materials in materials are the study of all these genetic material of particular organism or their gene structure or their study of their gene sequence are given in a genomics. So next is our main topic, that is what is metagenome, the complete set of genome from multiple organisms in a sample. A metagenome can include DNA from organisms, viruses, viroids and free DNA. Okay, so in a genome we study the genetic material, or in a genome, what is the meaning of this? As we see in the what is the meaning of genome, it is a genetic material of particular organism. Then what is the major difference in between the genome and metagenome? Though, so in a metagenome it is the complete set of genome from multiple organisms, not a single organism. It is the genetic material of a multiple organism in a simple. If you take the example, consider this is the bulk of the soil. Consider the bulk of the soil, and in this soil there are various type of microbes are present. If we isolate the genetic material of this entire or microbes, then this genetic material are called as metagenome. Okay, next is what is a metagenomics. Metagenomics is the study of the structure and the function of entire nucleotide sequence isolated and analyzed from all the organism in a bulk sample. Metagenomics is often used to study a specific community of organism and such as those residing on a human skin, in the soil or in the water sample. So, if we considered if we have to study the microbes which is present on our skin, if we isolate their genetic material in combine or in a bulk, this is called as metagenome. Or we study the particular sequence, or we study the sequences of the gene, or we study their genetic material, or we study the metagenome, or we study the genome in a bulk. This method, which deals with the study of genetic material in a bulk from specific organism, this method is called as metagenomics. In the metagenomics, we study the genetic material in a bulk form from the different organism or from the particular sample. This method is called as metagenomics. So these are the steps involved in the metagenomics. So these are the samples. As we seen, it is a large or small intestine of the humans. This is a bulk sample of the soil, and this is a water sample, that is a lake. Supposed to be, if we isolate the DNA from these three environment or from these three samples. First step is isolation of the DNA. Now this DNA are taken in a DNA library. So what is DNA library? DNA library is a collection of cloned DNA fragments that represent an organism genetic material. So DNA library, it is a place where we preserve the genetic material of an organism. Next step is DNA sequencing. So what is DNA sequencing? It is a laboratory technique that determines the order of the 4 bases that make up a DNA. So DNA sequencing simply means to study or to determine the sequences of the DNA, that is adenine, cytosine, guanine and thymine. This method of determining these 4 sequences are called as DNA sequencing. Next is sequence analysis and reconstruction. So sequence analysis and reconstruction meaning that the process of determining the order of nucleotide. It is a process of identifying or determining the order of nucleotides and in a DNA molecule, and the computerize them together, fragment DNA sequence to reconstruction the original one. This is in this step we analyze the sequence of the nucleotide of particular DNA or the group of DNA. Then fifth step is assemble genome sequence, and then tool software used to analyze the final genomic sequence. Which tools we use in a DNA sequence analyzing? That is a basic local alignment search tool, and also the various type of devices or tools used or software used to analyze the sequence of the gene. So this entire process or these entire steps come under the heading metagenomics, or all the steps are included in metagenomics. So next is what is the microbiome engineering. Microbiome engineering is a biotechnological process that involves altering the composition or activity of a microbial population to improve the function of an ecosystem. It has an application in human health, agriculture and climate changes. So microbiome engineering, it is a biotechnological process in which we improve the improve the particular microorganism. But how we improve this microorganism? By studying their metagenome, or by studying their metagenome, or by the metagenomics we are able to study their metagenome. And also if we by metagenomics we by done or by following the method of by using the method of metagenomics, we are able to know the sequence of this particular group of DNA sequence of this particular group of organism, and then which we are able to change the functions of particular genes of this organism, and this process is called as microbiome engineering. These changes in a microbial population is to improve the function of the ecosystem. There are various types of microorganisms are lived in an ecosystem, or the ecosystem is shelter for the various type of microorganisms. If we improve the functioning of microorganisms or particular group of microbials, so it is useful for the ecosystem. Also it is good for the human health, and it is useful in agriculture. And particular considered, this is the soil, and there are various type of microorganisms lived in the soil. And also this soil, it is the growing point of particular plant. If these microorganisms or these microbes are helped to this plant to grow fastly, or to grow without infections, then these microbes are useful in agriculture. And this to make this type of microorganisms are come under heading of microbiome engineering. And also useful in climate management. Here are some ways of microbiome engineering can be used. It is in human health, microbiome engineering can help to develop therapeutic and promote gut health. For example, fecal microbiota transplant can be used to treat the recurrent Clostridium difficile infections. So this type of this microorganism named as fecal microbiota useful to cure various type of infections in intestinal tract. In agriculture, microbiome engineering can improve the plant health and productivity, and help to address the environmental challenges like climate changes, desertification and salinity. So in agriculture, it is used to increase the productivity of particular plant, and to increase the growth or of particular plant, and also environmental challenges like climate change, desertification and salinity. Also useful in climate management, microbiome engineering can help in manage the climate changes. These are the some applications of the microbiome engineering. So the topic of introduction to the ethnobotany is over. Thank you.